these variables that you just presented, they sort of offer up a whole bunch of complexity, right? Like it sounds like you introduce complexity when you introduce all these variables. It's exciting, but I'm curious. I want to kind of stick with this for a minute. I want to Jeff, I want to welcome you to jump in on this question too. But
Sure.
As we explore all these variables and these different opportunities, how are we fundamentally maintaining or addressing the stability of RNA? I mean, we talk a lot about the stability of RNA in encapsulated form, and now when you introduce, I guess, additive technical approaches to an RNA in an encapsulated form, it's got to make that a more complex issue. So I guess let's start with the fundamentals. Like, what are the fundamentals of addressing stability?
Well, I'll just say a couple of things and I'll yield to Jeff. But I mean, the key is that mRNA itself is labile. It's easily degraded. So your delivery vehicle is all about protecting that mRNA from RNases in vivo and from degradation in vivo. Mm-hmm. So the first consideration for any type of a delivery vehicle is that it is protecting the mRNA. I mean, that's number one. You would not do any further work if you weren't getting adequate protection of the mRNA. So that's so all of these developments, that's the starting block, is protecting the mRNA. But then you're right, and we could discuss this for a while, it certainly does add complexities. Now, I can tell you that these groups working, say, in protein nanoparticles or DNA nanoparticles, they have expertise and been working in those areas for years. So they know how to make, they know how to test, they know how to look at, targeting. But, when you start doing things like anytime you start modifying any of these particles, you have to make sure they're still getting into the cell, they're getting released from the endosome, they're being able to express the protein in the cell. And then you're right, there could also be downstream manufacturing issues once you start to add any type of moiety or change your vehicle.
Mm-hmm. That's a fine segue to the CMC guy. Jeff, you're a CMC guy. So, Thomas says, we do some pretty cool stuff. Then we hand it off, it goes downstream, and let Atkinson deal with it.
Right.
Here you go, Jeff. Take this.
Yeah. So we'll step back all the way into the drug substance manufacturing, the mRNA, right? So some people are a lot of work has been done with modified nucleotides. So what's the best combination? What is it that helps us get the stability at the RNA level, right? So what is the best buffer to store it in? I mean, I think we've all kind of agreed that, storage of the mRNA at -70 is key, right? Some of them are good at a little warmer temperatures, and that's great. Is there something we can do there? And there may be, right? Some of that goes back to the design of the RNA. Then when we start getting into the drug product, it does start with those formulation buffers, right? Did you do what I'll call them the old school kind of your pre-formulation work, right? What are the buffers that you use to make sure that your mRNA is stable when you put it in and start the encapsulation process? And then is there anything we can do in that encapsulation process? There's always a lot of shear there. And that's inevitable in making these lipid nanoparticles. But is there anything that we can do to help reduce the stresses and strains on the mRNA as it gets encapsulated? And then it gets into some your final formulation buffer. Is that the right thing? Do you store it at -70, which is kind of where everybody's kind of settled right now? But there's a lot of work going on in conferences I've been to. There's people are talking, but nobody's really presenting, as far as I know yet, on lyophilizing these types of formulations, right? If we really want to move these into a more stable area, and when we start talking about access in markets that don't have the ability to have -70 storage, we need to get into something that's more typical for drug product storage, and that helps improve the stability, right? Like you're removing water, so hydrolysis kind of goes away. You need to look at the freeze thaw early. A lot of this is just early looks, right? Like really take a look at that stability of the drug product, both freeze thaw and long-term stability. As soon as you have a formulation, just start taking a look at it. Throw it in the freezer. It doesn't have, this doesn't have to be a GMP study, right? Like just throw it in there. Start seeing what makes a difference in your formulation.
Yeah.
And that helps us downstream in CMC to make sure that when we get it, it's already been through kind of that first cut of what helps it stay more stable.
Yeah. Yeah. Sticking with CMC just for a minute, Jeff, I'm reminded of a recent conversation I had with Dr. Nevin, I'm sorry, not doctor, but Nevin Elam at Resolute. He's the CEO and founder of Resolute. Now, this is an antibody company. They're late stage, and we were talking about sort of mitigation of late stage risk as you approach commercialization. And words that stuck out to me that he spoke were, when I did sort of post-mortem analyses, for lack of a better term, of regulatory disappointment in the antibody space, it almost inevitably tied back to some issue in CMC. Now I know this is obviously different from the antibody space, but I guess that's part of the question. Like, what do you know from previous exposure in the CMC space that you're applying now specific to mRNA, and what, if any, advances or developments in sort of the CMC environment in the mRNA space have you seen lately? And lessons learned. That's a multi-part question, I guess. But
right. I'm not sure if I'll remember all that, but we'll start talking, and you can kick me if there's something else that you want to talk about. But I think especially in the mRNA and LNP field, it's really trying to develop the platforms so that you're building this knowledge base, so that when you go to make the next product, you're already a leg up from where you were for the last one. Right. So the more you understand the platform, the better you're going to be the next time. The faster you can go, the more sure thing it is that you're going to make it to the end, assuming that it actually works. Right. It doesn't become a CMC issue. It now becomes does the product work? But there's, finding good partners, especially when we start talking about in the mRNA LNP space. There's a lot of people that are getting in that area. It's something that the manufacturing space is starting to free up a little bit, but there's still a fair lack of expertise out there. So it's choosing your partner carefully, and then when you get into scale up, which I think is probably what he was mostly talking about, were the difficulties in scaling up these kind of processes. It's not trivial. So a lot of the equipment manufacturers have gotten better in trying to develop their platforms so that they're more easily scaled, and so that's fantastic, and that's really helping out. But then I think the methodologies, the methods, and the instrumentation, there's also a lot of work going on there so that we can better characterize these kind of products. I think that there's still a long way to go. There's a lot more analytical work that needs to be done to try to figure out exactly what we're getting. Right now, we're kind of depending on, well, if we do the same thing every time, we're going to get the same product, which I'd say is mostly true, but do we understand exactly what that product is? And I'd say in many important aspects we do, but there's a lot that we don't know. I'm not suggesting that these become release criteria, but characterization criteria so that we understand what it is and can utilize that information, not just for the product we're working on now, but for the one that we're working on next year or the year after or the year after.